All right, so this next phase of the cycle, I really want to focus in on that M phase, the mitotic phase, mitosis. You will need to know what happens during each phase of mitosis, and tell me about what the chromosomes are doing. You guys know, again, I enjoy my essays. This seems like one of them. There is a difference between mitosis and mitotic cell division. What you've probably learned about previously is mitotic cell division, where you're going to divide the whole cell. Mitosis is a part of mitotic cell division. This mitotic cell division will produce two identical daughter cells, two cells that are exactly the same as the parent, genetically identical. Yes, there is a difference between mitosis and meiosis. Mitosis produces identical daughter cells. Meiosis is going to produce genetically distinct daughter cells. We're going to get into meiosis in the next lecture, but don't worry, we're going to get there. I'll teach you all how it works. It's a very interesting and a very important concept when we're getting ready to study genetics. Mitotic cell division will allow us to not us, sorry, didn't mean to say us, them. Gosh, it really is. I'm talking about me as an amoeba. It will allow other organisms like bacteria, not even bacteria, like amoeba, like paramecia, to create an exact copy of themselves. They don't need to mate with somebody else. They can just split. When a cell begins to divide, it creates two identical chromatids, right? We talked about this in the earlier lecture. You have a single chromosome. That chromosome is going to reproduce itself. You're going to have this happen. When does this happen? What phase do you copy your DNA? S phase. S phase during interphase, right? So you double the amount of DNA present. They're going to be connected at that centromere. And what surrounds the centromere? The kinetochore, that belt around it. I know, that's why I called on you. We use the kinetochore to help organize the chromosomes. It's a belt surrounding them. Attaching to the belt are microtubules. Now, what have microtubules been used for in the class? Transportation, right? In this case, they're going to transport chromosomes from one place to another. To do that, they can't actually physically bind to the centromere, so they bind to the kinetochore around it. And that allows the chromosome to get pushed or pulled. It's sort of like if you've ever pulled somebody by the belt, they have to go where you pull them. Now, there are going to be several steps to mitosis. Oftentimes, people include interphase. I tend not to. Interphase is outside of the mitotic, out of mitosis. But a lot of time, people include interphase and then prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis is occurring throughout. I'm going to go through each of these step by step. So if you didn't get them listed in order, don't worry. The first step is interphase. Good news, we did a whole lecture on this. If you want, you can go and review the whole thing. It was the one right before this one. During interphase, the genetic material is replicated. The cell grows. The cell checks itself out. At this point, all of the genetic material is chromatin. You don't have chromosomes yet. They're not condensed. So G1, you get bigger. S phase, you copy your DNA. G2, you're going to check yourself out and prepare proteins for mitosis. So this is most of the cell's life. As the cell enters mitosis, if it's past all the checkpoints, and it goes into mitosis, you enter into the first part of mitosis, which is called prophase. Pro meaning first or before. So the first phase. During prophase, first thing that happens is the nuclear envelope begins to dissolve. That is, that nuclear membrane, you know how it's double membrane bound? It goes away. It gets packed into vesicles, shipped to different parts of the cell. That frees the genetic material to be in the cell as a whole. The chromatin starts to condense and become chromosomes. And the centrioles, otherwise known as the centrosome, you can use them interchangeably. Two centrioles make a centrosome. Begin to produce what's called the mitotic spindle. That is, they produce microtubules. They provide an attachment point for microtubules. So the three things happening: the nuclear envelope goes away, the chromosomes condense, and the microtubules begin to form at the centrosome. No more nucleus, chromosomes appear, and the microtubules begin to form. That's all step one, prophase. So then we enter into pro-metaphase. Again, what does pro mean? First or before. So this is first before metaphase. The whole thing that happens in this phase, and it's a vital step, is the microtubules attached to the kinetochore. That's it. You attach the microtubules to the kinetochore surrounding the centromere. You've definitely got your really thick chromosomes. You're connecting everything together. Moving on. Once you've connected the microtubules to the centromeres, you can start pushing or pulling the chromosomes into position. Now, the way you can imagine this working is, imagine two fishermen standing on opposite sides of a room. I guess not a room because there's fishermen, so I don't know, opposite sides of a pond. In the middle of a pond is a tire. If they've both caught the tire on their hook, if one of them pulls the tire toward them, the tire moves in this toward that one person, right? At the same point, the other person has to be letting off string. Does that make sense? To move the tire to the other end, the second person needs to start reeling in, and the first person starts to let go of the string. Between the two of them, they can position that tire wherever they want in the pond by either pulling in or releasing the tension. That's what the microtubules are doing. They've latched on, and by either growing in one direction and shrinking the other, or vice versa, they can start moving those chromosomes around the cell. So what they do is they move the chromosomes until they're even between the two centrosomes. That's where they're lined up at what's called the metaphase plate. They're lined up in the center of the cell. So metaphase is lining up the chromosomes at the metaphase plate. Next up is anaphase. Ana means without. Here we're going to pull these things apart. You're going to take the two sister chromatids and the chromosomes and rip them apart from each other. They get pulled to either end of the cell. So you start off with, a chromosome. That chromosome is going to double until you have two sister chromatids. You're going to attach these to a microtubule, and that microtubule rips them apart. So anaphase is you're pulling these two chromatids apart. It's important to note that at this stage, you have individual chromosomes again. These are not called chromatids. These are called chromosomes. So that's anaphase. Telophase is where the nuclear envelope is going to reform. The chromosomes begin to decondense. Effectively, we're getting right back to where we started. You've got two distinct nuclei now, each of which have the exact same set of instructions in them. So you have two identical daughter nuclei at either end of the cell. So telo, it means, I think it's distant, far apart. You're looking at two nuclei that are far apart. The nucleus reforms, the chromosomes decondense. We start off at exactly where we started. We end at exactly where we started in G1 with the nucleus. A nucleus that has a bunch of chromatin in it. And that's why it's a cycle. This telophase will then lead back into G1. Throughout the whole process, cytokinesis is occurring. Cyto meaning cell, kinesis meaning movement. So the cell is splitting. It begins to split in prophase and finishes splitting in telophase. In animal cells, a ring of actin filaments wraps around the whole cell. Now, what's actin good for? What does it cause? Actin causes actions, right? So what you have is a belt of actin. And as mitosis goes on, the belt gets tighter and tighter and tighter until eventually the two cells split apart. This is exactly analogous to how vesicles form. You've seen this before in the Golgi apparatus, except it's happening on a much larger scale. It's happening to the whole cell. So cytokinesis will cause this cleavage furrow to form, this sort of bump in there, and that gets deeper and deeper and deeper until the two cells are split. This cannot happen in plant cells. What's the big difference between plant and animal cells? The cell wall. Cell wall's too hard. Actin filaments can't do this. So instead, in plant cells, you're going to have individual, what's called a cell plate form. The Golgi apparatus begins to ship proteins to the center of the cell. These proteins are usually used to build a cell wall. So what happens is you start to build teeny tiny bits of a cell wall that begin to unite until they form a plate, and then that plate goes all the way across to form a wall. It's almost like if you were to, when you were younger, did you ever try to dam up a stream? All right, when you tried to dam up a stream, what did you use? Rocks, anything you could find, right? Usually the bigger stuff, but rocks. You take a rock, you put it in the middle of the stream. What happens? What? It stays in the water, and the water keeps going around it, right? One rock doesn't do much. So what do? You get more rocks. You take a bunch of rocks, you start dropping them in the same place, and eventually you build a wall. Eventually, the whole stream gets blocked, and that's what's happening here in this plant cell. You're dropping off individual proteins, one by one, that unite to form a bigger wall. So that's in plant cells. Animal cells, they'll have a cleavage furrow, but plant cells will end up forming the cell wall. For the inevitable question, what's the difference between cytokinesis in plant cells and animal cells? So mitosis ensures genetic consistency. In us, I got it right that time, multicellular organisms, that's really important because my cells need to recognize each other as the same. Otherwise, they're going to attack themselves. Otherwise, they're going to try and defend themselves from what should be their team. It'd be like trying to play football with no jerseys. You just got to figure out who's on your team and just, whoever gets the ball, just hit them. What mitosis does is allow cells to be able to recognize each other. It also allows us to work by the same instructions. Have you ever been on a team where you had two people trying to be the leader? And I've seen you guys in lab. I know how this works. You have two people trying to be the leader, and sometimes they give conflicting information. Does that make sense? There's conflicting problems. And when that occurs, things aren't very efficient. One set of instructions allows you to have more efficient communication. And that's an armadillo. I found a good picture of an armadillo. It's like an armored rat, pretty much. It's a rat ready for battle. But cool thing about armadillo is armadillo are all born as quadruplets. You guys ever seen an armadillo? If you've ever lived have you ever lived in Florida or Texas, these guys are all over. They're pretty much roadkill everywhere. I know. But if you ever happen to be in Florida and hit an armadillo, don't worry. because it turns out there's 3 more exactly like it out there somewhere. So there we go.